So in this session, we'll deal with the further development of embryo. In earlier sessions, we have seen the second week and third week development. Now we'll be seeing from 4th to 8th week development. So the embryonic period, also called as organogenic period, so it extends from 4 to 8 weeks. So ectoderm, endoderm, and mesoderm, everything is changing, and they are assigned the fate for further development. The germ disc gets folded to form identifiable shape of human embryo. So events in embryonic period are grouped like this, like ectodermal differentiation, mesodermal differentiation, embryonic folding, and endodermal differentiation. So the teratogens, if they act during this embryonic period, they have the maximum capability to produce congenital anomalies. So at the beginning of the third week, central part of the ectoderm overlaying the developing notochord thickens to form neural plate or neuroectoderm. We have already seen in the third week development and also in the development of nervous system in detail about this. Remaining part of the ectoderm is called surface ectoderm, and this particular part will be called as neuroectoderm. So the neural plate goes to form peripheral neural folds and the central depressed area called neural root. Neural fold starts fusion in the center cervical region. Fusion extends craniocaudally to form neural tube. Cranial opening end of the neural tube called anterior neuropole goes by 25th day. The caudal open end of the neural tube called posterior neuropole goes by 27th day. You can see this is the cranial neuropole, this is the caudal neuropole, this is the closure of the neural tube. You can also see the somites here. So cells at the margin of neural plate form the neural crust. The neural tube gets separated from surface ectoderm, whereas neural crest cells come to lie between the ectoderm and the neural tube. The cephalic part of the neural tube shows dilatations as forebrain vesicle, midbrain vesicle, hindbrain vesicles, whereas distal part of the neural tube forms the spinal cord. All these have been discussed in detail in the development of the nervous system. So neuroectoderm form entire central and peripheral nervous system and autonomic ganglia. So here you can see this is the yolk sac. You can see these are the somites. You can see the anterior neuropore and you can see the posterior neuropore. You can see here there is a connecting stalk. So you can see the formation of the head fold and tail fold. You can see the complete formation of the connecting stalk. It is the umbilical cord. You can see the vital line duct, umbilical cord, umbilical blood vessels. You can see the allantois. You can also see the limb bridge. You can see the development of pharyngeal arches. You can see the lens placode. You can see the otic placode for ear, lens placode for development of eye. You can see the arrangement of somites which develop from the paraxial mesoderm. The surface ectoderm shows depression at buccopharyngeal membrane and cloacal membrane. So at these two depressions, ectoderm is firmly adherent to the endoderm. There is no mesoderm. So buccopharyngeal membrane, also called oropharyngeal membrane. In third week, it lies cranial to precaudal plate. On development of the head fold, it comes to lie between forebrain vesicle and the pericardial bulging. So at the beginning of the fourth week, buccopharyngeal membrane ruptures to form communication between the amniotic cavity and primitive bed. That will be the stomodium. So cloacal membrane divides into anterior urogenital membrane and posterior anal membrane with the development of urorectal septum. You can see these are the derivatives of surface ectoderm. The surface ectoderm forms the following structures like epidermis, hair and nails, sebaceous glands, sweat glands, olfactory pit, optic vesicle and lens vesicle, otic vesicle, branchial clefts, Rathke's pouch, so which helps in formation of this thing like pituitary gland, epithelial lining of the cheek, gum, teeth, enamel, root of the mouth, then nasal cavity, paranasal air sinuses, salivary glands, mammary glands, and pituitary gland. So you can see the mesoderm, which is differentiated into three important structures. You can see from medial to lateral side, the paraxial mesoderm, intermediate mesoderm, and lateral breadth mesoderm. You can also see the presence of neural crest cells here. See the different levels at which the sections are being projected here. You can also see here, this is the intraembryonic coelom. So this will be the somatopluric and this will be the splanchnopluric. Splanchnopluric lateral plate mesoderm is more towards the yolk sac and the somatopluric layer is towards the amniotic cavity. You can see the clear differentiation, diagrammatic representation. The intermediate mesoderm is being divisible into paraxial mesoderm, which lies on either side of the axis, intermediate mesoderm and lateral plate mesoderm. You can see the paraxial mesoderm dividing into somites, and you can see the intermediate mesoderm, and here you can see the lateral plate mesoderm. The lateral plate mesoderm is divisible into the somatopluric layer towards the amniotic cavity and the splanchnopluric layer towards the yolk sac. You can see this is the notochord, and on the other side of the notochord you can see the paraxial mesoderm. Just lateral to paraxial mesoderm you can see the intermediate mesoderm. So by the end of the third week, primitive streak starts regressing. The mesodermal cells that are migrated from primitive streak start forming rod-shaped on either side of the notochord. So condensed mesodermal rods are separated by shallow longitudinal grooves into three parts. Paraxial mesoderm lies on either side of the notochord. Intermediate mesoderm lies lateral to paraxial mesoderm. Lateral mesoderm is the lateralmost part of the mesoderm, as I've shown in the earlier diagrams. So paraxial mesoderm, name itself speaks, situated on either side of the notochord, extend from precaudal plate to primitive streak, form a series of rounded oval-like masses called somatomeres. The somatomeres further form discrete block of segmental mesoderm called the somites or metameres. You can see here. So this is the somite. You can see the dorsal dermatome. You can see the ventromedial sclerotome. You can see the intermediate xylem in the lateral plate mesoderm. And this is the intermediate mesoderm. So lies between paraxial mesoderm and lateral plate mesoderm. It forms kidneys and sex glands, ovaries and testes. Then the lateral plate mesoderm extends from intermediate mesoderm to extremity mesoderm. So cranially it is continued with pericardial bar. Pericardial bar is mesodermal condensation cranial to buccopharyngeal membrane. Small cavities appear in lateral plate mesoderm, which fuse to form the intraembryonic xylem. Similar cavity develops in pericardial bar called pericardial sac. So intraembryonic xylem divides lateral plate mesoderm into two layers: somatopluric layer. The parietal layer lies in contact with ectoderm. Splanchnopluric layer, visceral layer, lies in contact with endoderm. Continuously growing intraembryonic xylem separates somatopluric and splanchnopluric layers laterally and develops communication with extraembryonic xylem. Septum transversum, it is a cranial mesodermal horseshoe-shaped bar connecting somatopluric layer with the splanchnopluric layer. So it later forms the diaphragm. So pericardial sac communicates with intraembryonic xylem to form inverted U-shaped tubular passage. The coelomic cavity forms pericardial, pleural and peritoneal cavities. So lateral plate mesoderm, the somatopluric layer, parietal layers of peritoneal, pericardial and pleural cavities. It also forms the dermis, pectoral and pelvic girdles, skeletal elements of the limbs. Splanchnopluric layer, so it's a visceral layer of the pericardial, peritoneal and pleural cavities are formed from this. The musculature and connective tissue of the gut, respiratory tract and heart. All these develop from the splanchnopluric layer. You can see here paraxial mesoderm, sclerotome which forms ribs and vertebrae, myotome which forms skeletal muscle, dermatome which forms the dermis of the skin. Intermediate mesoderm, kidney and sex glands, testes and ovary. The lateral plate mesoderm, the splanchnopluric layer, connective tissue of the gut, respiratory tract and heart. The somatopluric layer, parietal layers of the peritoneum, pleura, pericardium, skeletal elements of the limbs. So paraxial mesoderm, intermediate mesoderm and lateral plate mesoderm. These are the three parts of the intraembryonic mesoderm. So paraxial mesoderm on condensation forms centromeres that later on segmentation form somites from the precaudal plate to the primitive streak. So it extends, these somites extend from precaudal plate to the primitive streak. The proximal somatomeres underlying the developing brain, rostral to the otic vesicle, remains unsegmented and do not form somites. These form striated muscles of the face, jaw and throat. The precaudal plate mesoderm form preotic somites that later form extensive muscles of the eye. So first pair of somites appears on the 20th day on each side of the cranial end of the notochord. So first appearance is on the 20th day. Somites continue to appear from 20th day to 30th day in craniocaudal direction. These are the somites. So in human, 42 to 44 pairs of somites form. Occipital somites 4 pairs, cervical 8 pairs, thoracic 12 pairs, sacral 5 pairs, coccygeal 8 to 10 pairs. Many coccygeal pairs of somites eventually disappears till it reaches final count of 37 pairs of somites. Counting of somites. So first somite pair appears on day 20 and further 3 pairs of somite get segmented each day until the end of the 5th week. And somites gives idea about the fetal age. You can see the anterior neuropore, the posterior neuropore, you can see the somites extending. So if we see approximately from day 20 to day 30, so on day 20 there are 1 to 3 somites, 21st day 4 to 7, 22nd 7 to 10 somites, 23rd 10 to 13, 24th 13 to 16, 25th day 16 to 19, 26th day 19 to 21, 27th day 21 to 24, 28th day 24 to 27, and 30th day 34 to 35 somites. So somite is differentiated into a sclerotome, so ventromedial part forms ribs and vertebrae. Myotome, its middle part forms skeletal muscles. Dermatome is the dorsolateral part and forms the dermis of the skin. Small cavity of somite is obliterated by proliferation of cells. Each somite is supplied by a single spinal nerve. Some authors consider dermomyotome instead of separate dermatome and myotome. The occipital somites and preaortic somites participate in the formation of the base of the skull. So somites, these are the blocks of paraxial mesoderm. The formation, paraxial mesoderm, somatomers, somites, the first appearance in the occipital region and extends in craniocaudal direction. Preoccipital 3, occipital 4, cervical 8, thoracic 12, sacral 5, coccygeal 8 to 10. So day 20, 1 to 4 pairs, on day 20, 24 to 27 pairs. Daily 3 pairs are getting added up. So structure, it is sclerotome, ventromedial sclerotome, dorsolateral dermatome, intermediate myotome. Sclerotome gives us to ribs and vertebrae, myotome to skeletal muscles, dermatome, dermis of the skin. The preoccipital somites gives rise to extraocular muscles of the eyeball, and the occipital somites the muscles of the tongue. You can see here there is a neural groove. You can see the sclerotome and dermomyotome of the somite, intermediate mesoderm, the lateral plate mesoderm, and the intramembranous xylem. So cranial to caudal, these are the following structures which we will be seeing: the septum transversum, pericardial sac, buccopharyngeal membrane, notochord, neural tube, primitive node, primitive steak, cloacal membrane. I'll show you this in further diagrams. So a small note on vasculogenesis. So vasculogenesis is process of formation of a new vessel. Angiogenesis is sprouting of vessels from already existing vessels by endothelial budding. So at the beginning of the third week, formation of vasculature begins in the extraembryonic mesoderm of the yolk sac, connecting stalk and chorion. So on day 17, the mesenchymal cells differentiate into hemangioblasts. Hemangioblasts aggregate to form cluster of cells called blood islands. So hemangioblasts differentiate into two cell lineages as follows. The primitive hematopoietic stem cells form erythropoietic cells, megakaryocytes, and primitive macrophages. Endothelial precursor cells form epithelial line with primitive endothelium. So primitive hematopoietic stem cells and endothelial precursor cells. These two are derived from the hemangioblasts. So small cavities appear in the blood islands to form lumen of the primitive capillaries. Primitive hemangioblast islands fuse to develop capillary networks, vasculogenesis. So endothelial precursor cells sprout into adjacent mesenchyme and fuse with adjacent hemangioblastic islands and develop new capillaries, angiogenesis. So by the end of the third week, yolk sac, connecting stalk, and chorion get completely vascularized. So yolk sac is the first supplier of the blood cells up to 60 days. Later, liver, spleen, thymus, and bone marrow take over the hematopoietic function. Bone marrow take over the hematopoietic function. So you can see the vasculogenesis beginning with the formation of the hemangioblast, differentiating into hematopoietic stem cells and endothelial precursor cells. So endothelial precursor cells forming the endothelium, hematopoietic stem cells forming the blood cells. So mesenchymal cells differentiates into hemangioblasts, hemangioblasts into hematopoietic stem cells and endothelial precursor cells. As you can see, they are forming blood lines, cavities, and even of vessels. Hematopoietic stem cells giving rise to erythrocytes, megakaryocytes, and macrophages. Endothelial precursor cells giving rise to primitive endocardium and also the endothelium. Now coming to the actual topic, the foldings of embryo, which is most important. Cells lying in the central part of the germ disc multiply rapidly than that at the periphery. So overgrown central part result in the folding of germ disc and forms primitive cylindrical embryo. So due to overgrowing tissue, the embryo shows folding in the median plane, cephalocaudal folding, and lateral plane, lateral folding. Cephalocaudal folding can be studied as cranial head fold and caudal tail fold. Amniotic cavity enlarges to enclose ventral surface of the embryo, and amniotic membrane forms a tubular covering around the connecting stalk. So all the folds converge on the ventral surface of the embryo to produce hourglass constriction of the yolk sac. So part of the yolk sac trapped within the embryo form primitive gut, and the protruding part of the yolk sac extremity part forms the umbilical vesicle. Vital intestinal duct temporarily connects primitive gut with the umbilical vesicle. So part of the primitive gut, cranial to vital intestinal duct, is foregut. Caudal to it is hindgut, whereas the part of the gut communicating with the vital intestinal duct is the midgut. So overgrowing germ disc folds around cranial end of the notochord to form head fold. So foregut, this is the trapped part of the yolk sac in the head fold. Ventral relations of the foregut are the buccopharyngeal membrane, pericardial sac, and septum transversum. Dorsal relations of foregut are the notochord and brain vesicles. Cranial relation of the foregut is rapidly growing forebrain vesicle. Due to head fold, the buccopharyngeal membrane come to lie on the ventral surface of the embryo and becomes cranial to the pericardial sac and septum transversum. During the fourth week, buccopharyngeal membrane ruptures. Foregut is separated from pericardial sac by cardiogenic plate. Septum transversum is the mesenchymal tissue that lies caudal to the pericardial sac. Septum transversum forms fibrous pericardium, part of the diaphragm, and part of the ventral mesentery of the foregut. So let me show you here. You can see this is how a trilaminar germ disc appears. You can see this is the amniotic cavity, this is the yolk sac, you can see the allantois, this is the connecting stalk. You can see the formation of the head fold here. You can also see the formation of the tail fold here. See the head fold and tail fold converging on the ventral aspect of the embryo. So initially, if you observe, this is the septum transversum, this is the pericardial cavity. You can see the primitive heart. So cranial most structure is the septum transversum. Due to, and the caudalmost part is the allantois. So due to formation of the head fold and tail fold, you see the changes in the position. The septum transversum has come to lie at its final level, that is the future diaphragm place, and you can see the pericardial cavity has become cranial to the septum transversum. And you can see here this is the buccopharyngeal membrane. Here you can see this is the cloacal membrane, and you can see the change in position of cloacal membrane also. So the cloacal membrane has come from dorsal aspect to the completely ventral aspect. You can see the position of change in the allantois. So you can see this is the yolk sac. So this particular part, this will form the vital intestinal duct, this forms the foregut, this forms the midgut, and this forms the hindgut. This forms the umbilical vesicle. Overgrowing germ disc also fold ventrally around the caudal end of the notochord to form tail fold. The tail fold produces the following changes. So hindgut trapped part of the, this is the trapped part of the yolk sac in the tail fold. Dorsal relations, notochord and neural tube. Caudal relations, degenerating primitive node and streak. Allantois communicates with the hindgut. Bilaminar cloacal membrane lies caudal to the connecting stalk. So you can see here, this is the primitive streak. You can see the cloacal membrane. You can see the allantois communicating with the hindgut. See the initial position, see the final position after the formation of the tail fold. Caudal to the hindgut, a bilaminar area of the germ disc is called cloacal membrane. So you can see here. So this is the cloacal membrane, caudal to the germ disc. The site of cloacal membrane, ectoderm shows a depression called ectodermal cloaca. Post-aligned part of the hindgut dilates to form endodermal cloaca. So cloacal membrane is divided into ventral urogenital membrane, dorsal anal membrane. With the advent of the urorectal septum, it will be dealt while dealing with the development of the hindgut. Both urogenital and anal membranes rupture in the 5th week. Lateral folds. Overgrowing central part of the germ disc forms two lateral folds, right and left. Formation of the lateral fold produces following changes. The amniotic junction come to lie on umbilical cord. Formation of the midgut and vital intestinal duct and umbilical vesicle. Formation of dorsal mesentery of the gut. Splanchnopleuric intraembryonic mesodermal layer for dorsal mesentery. Peritoneal cavity. The somatopleuric intraembryonic mesoderm fuse ventrally in the midline to convert intraembryonic coelom into peritoneal cavity. You can see here the somatopleuric layer coming like this on both the sides, and it will try to enclose it completely. You can see the intraembryonic coelom. Here it is the beginning. Here you can see the intraembryonic coelom communicating with the extraembryonic coelom. You can see the amniotic cavity. It is trying to cover the entire yolk sac by forming the lateral fold. At the same time, the somatopleuric layer of the lateral plate mesoderm is also covering like this. You can see here the amniotic cavity trying to take some part of the yolk sac to inside. So this is the hourglass construction of the yolk sac. So this part forms the umbilical vesicle, this part forms the primitive gut. So now you can see completely the somatopleuric layer here is actually forming the body wall. You can see this is the open part where you are seeing the vital intestinal communication between the midgut and the umbilical vesicle. You can see here in the closed part, you can see here. So this is the hindgut. So this part is completely surrounded by the splanchnopleuric layer, and this is the somatopleuric layer of the intraembryonic mesoderm. So this is the peritoneal cavity. You can see these are the lateral folds which have closed. Thus head fold from the cranial side, tail fold from the caudal side, and the two lateral folds. Everything converges towards the umbilicus. Foldings of embryo, rapid growth of central part of embryo, 4 folds, head fold, tail fold, and 2 lateral folds. So flat trilaminar germ disc is converted into cylindrical embryo. Hourglass construction of the yolk sac, as I've shown in the diagram. You can see here, this is the hourglass construction of the yolk sac. Gut tube. So this is the gut tube being formed. Vital intestinal duct. So this is the connection between the gut tube and the umbilical vesicle, and the last one is the umbilical vesicle. Formation of the connecting stalk is the umbilical cord. Formation of the tail fold moves the attachment to the connecting stalk from dorsal end of the germ disc to the ventral aspect of the embryo and limit it to the umbilical opening. Structure of the connecting stalk in the 5th week consists of the vital intestinal duct, remnants of the yolk sac, extraembryonic mesodermal connective tissue that forms the Wharton's jelly, which is nothing but the connective tissue of the umbilical cord and wheels of the umbilical cord, umbilical arteries and vein. Continuous stalk is the site of amnioectodermal reflection, continuation of the intraembryonic and extraembryonic coelom. This coelom continuation persists up to 10th week. So you can see here. So this was the bilaminar germ disc. So now you can see here the extraembryonic mesoderm. You can see the amniotic cavity completely encircling here with the formation of the lateral folds, taking a part of the yolk sac inside, forming the gut tube, foregut, midgut, and hindgut. You can see this is the umbilical vesicle, this is the vital intestinal duct. You can see the formation of the umbilical cord. So what is happening with the endoderm? So this endoderm, that is the yolk sac, you can see this is the vital intestinal duct. Here will be the umbilical vesicle. This is the part which is retained within the embryo, is forming 3 tubes, that is the foregut, midgut, and hindgut, all together called as primitive gut. Derivatives. So endoderm of the foregut, lining of the epithelium of pharynx, esophagus, stomach, duodenum up to the ample of the water, tongue, floor, and mouth. The lining epithelium of respiratory system, auditory tube, tympanic cavity. Parenchyma with the tonsil, thyroid, parathyroid, thymus, liver, and pancreas. Midgut, lining epithelium of distal part of the duodenum, jejunum, ileum, cecum, appendix, ascending colon, right 2/3 of transverse colon. Hindgut lining epithelium of left one third of the transverse colon, descending colon, sigmoid colon, rectum, anal canal up to mucocutaneous junction. Lining epithelium of urinary bladder, urethra and vagina, and the parenchyma of the prostate and bulbourethral glands. So these are the derivatives of the foregut, midgut, and hindgut. These are nothing but the endodermal derivatives. Now, if you see the week-wise changes, to consolidate, in the fourth week, there is increase in somite number, formation of 3 brain vesicles of the neural tube, formation of the head and tail folds, 21st days and later, heart starts beating, 24th day, first pharyngeal arch appears, 26th day, 3 pairs of pharyngeal arches, 26th-27th day, rudimentary forelimb bud, 28th day, rudimentary hindlimb bud, and formation of the prominence of forebrain vesicle, otic pits, lens, placodes appear. These are the ectodermal thickenings. Fifth week, rapid growth of facial and head prominences, rapid growth of second pharyngeal arch, growth of the limb buds, formation of the alar and basal lamina of the neural tube, appearance of olfactory placodes, maxillary and frontonasal process, which will be dealt in the development of face. Sixth week shows spontaneous movements, growth of brain vesicles, appearance of nasal process, buccopharyngeal membrane ruptures, development of appendix, cecum and spleen, differentiation of the limb digits begins. Differentiation in lower limb starts 4 to 5 days later than upper limb. Umbilical herniation is common during this week. 7 to 8 weeks, development of face, external ear, so auricular hillock and eye. So well-defined limbs and digits, development of metanephric kidney, testis and ovary, differentiation of genitalia. During 8th week, coordinated limb movement occurs, primary aspiration center starts appearing, and caudal tail-like eminence appears. Thank you.